There are three main types of topo-based cloning strategies. The topo cloning that uses blunt ends. The modification to classic topo is TOPO TA, which uses sticky ends. Third is directional topo, which uses both a sticky and a blunt end. In case you're not familiar, the topo is a shorthand for the enzyme topoisomerase. This is a very popular strategy to clone PCR product, and it bypasses the need for restriction enzymes and ligases that we saw in classic molecular cloning. You will understand topo cloning if you understand the basics of topoisomerase. In the most simple form, a topoisomerase binds to a supercoiled DNA, makes a cut in one of the DNA strands, and subsequently religates the cut. Through this simple cut in the supercoiled DNA, the DNA undergoes relaxation. Following the religation of the DNA and relaxation of the DNA, the topoisomerase walks away. In topo cloning, the topoisomerase that is used is called Vaccinia topoisomerase. Vaccinia is an ancestor of the smallpox virus. You don't have to know this, but Vaccinia topo is a type 1 beta class topoisomerase. The important thing to know is that it has two binding and cleaving sites. Think of them as DNA motifs that attract the topoisomerase to the DNA. CCCTT is one of the two binding sites. If present in a double-stranded DNA context, the Vaccinia topo will recognize this site and make a cut after the last thymine in the CCCTT sequence. The second sequence of specificity is TCCTT. Same logic here as well. The cut is made after the last T. If you recall our simple diagram here, I did say that the cut is made only in one of the DNA strands. This cleaving activity is called a nicking activity since only one of the strands is cut. This is very different from restriction enzymes, where a DNA sequence is recognized, but the cut is made on both DNA strands, which actually splits the DNA into two parts. Generally, the CCCTT sequence is most used in topo cloning, so I will restrict our discussion to just this one. There's a little bit of biochemistry you must understand regarding the nicking or binding cleaving activity of the topoisomerase. Let's zoom into this nick location. Here's the top strand of the DNA with its backbone. In the active site of topoisomerase, there is a tyrosine amino acid. From the N-terminal end, it is the 274th amino acid. The free hydroxyl of the tyrosine attacks the DNA backbone between the T and the N base. It specifically attacks the phosphate. This is a nucleophilic substitution reaction. This is what cuts or nicks the DNA. When the substitution reaction is complete, the phosphate gets connected to the tyrosine 274 of the topoisomerase. If you annotate the direction of this DNA, you notice it is a 3' phosphate connected to a tyrosine. So we call it a DNA 3' phosphotyrosyl enzyme linkage. This means that the next base, the base N, is cut off from the T base. And the 5' end of the base N has a hydroxyl because the phosphate is on the 3' end of the base thymine. In a way, this reaction has dephosphorylated the base N during this cleaving step. Now, if we revisit the cut and ligation step of topoisomerase, we can add an intermediate step containing a DNA enzyme linkage. But why, or rather, how is this step an intermediate step? If you look closely, the free hydroxyl of the 5' end here is similar to the free hydroxyl on the topoisomerase. So this is also free to attack the phosphate in this DNA enzyme linkage. This will be yet another nucleophilic substitution. When this second reaction is complete, the two bases are religated back through the phosphodiester linkage. We have now come full circle to the original stage. Just to reiterate, the first attack is from the enzyme to the DNA. This is a nucleophilic substitution. The second attack is a DNA to DNA, which again is a nucleophilic substitution. And this way, the intermediate stage resolves itself and the DNA ends are able to ligate without any need of DNA ligases. This intermediate step is actually everything. Hopefully you understand the basics, so let's get into topo cloning. First type is the blunt topo cloning. All topo cloning vectors rely on stabilizing or artificially creating a stable intermediate stage. All topo vectors are linear. The TOPO Blunt vector is meant for cloning DNA. It is not an expression vector. The ends of the TOPO Blunt vector have the topoisomerase recognition sequence CCTT. This is one continuous DNA, but the ends are not connected. Hence, it is a linear vector. At this recognition site, the intermediate stage is stabilized, meaning that the T is connected to the topo on both ends of the vector. This is the 3' phosphotyrosine linkage we discussed earlier, which occurs at the base T. That is the vector, but what about the insert? Note that the vector is blunt, meaning that the insert must also be blunt. If you get your insert from a digest, you must then dephosphorylate the insert using a phosphatase. You may be wondering, why is dephosphorylation required? If you recall the intermediate step, the 5' end needs a hydroxyl to perform the substitution. TOPO cloning is typically meant for PCR inserts. Typically, PCR amplicons are blunt if you use some proofreading DNA polymerase. Normal PCR primers are not phosphorylated, so the 5' end of a blunt PCR has this same dephosphorylated biochemical structure. So here is the ideal form of the insert. The ends have a free hydroxyl, no phosphate, and they are blunt. The hydroxyl can perform nucleophilic substitution by attacking the phosphate in the vector. The insert will replace the enzyme linkage and the phosphodiester bond is created at both 5' ends of the insert. But wait, we have these two 3' ends and the 5' ends of the vector. They are not yet ligated. So this is a partially closed vector. It is circular but not fully closed. Once inside the bacteria, and the remaining nicks can be fixed. So this way, a linear vector can be turned into a circular vector. There is a small catch. Say the insert has two locations X and Y. And it is also possible that this hydroxyl attacks this phosphate and the other hydroxyl attacks this phosphate, right? So in the first case, the X to Y orientation can be cloned. In the other case, the insert can be flipped and the Y to X orientation gets inserted. So depending on which hydroxyl and phosphate combination is used, you can get two types of final vectors. This is also the principle of blunt restriction enzyme cloning, that in the end you will have to screen for your desired vector. There is one problem with this, a high probability problem. Both the 5' ends of the vector also have free hydroxyls in them, which can also attack the phosphate, and when it happens you close the linear vector by itself. How do you deal with this problem? The easiest way is to get rid of hydroxyls from the ends of the vector. The alternative, and this is the most widely used strategy, is to have the TOPO cloning sites in the middle of a gene called CCDB. If a vector closes itself, the gene is reconstructed and it becomes active. Once this self-ligated vector is inside the bacteria, the product of the active gene will kill the bacteria. Alternatively, if you have inserted a piece of DNA in the cloning site, then CCDB is inactive, and those bacteria will survive. Second type is TOPO TA cloning. This vector has sticky overhangs. It is missing the complementary 3' adenine. So this is a T overhang vector. What about the insert? Well, it needs to be sticky. You can get them from a sticky digest like XCM1 or ADH1. Like we saw before, it then must be dephosphorylated for the nucleophilic substitution to work. These sticky digests give you an A overhang. What about PCR amplicons? If you do a PCR with a non-proofreading enzyme, like the Taq polymerase, they can add an adenine nucleotide at the end of your PCR product. This is a template-independent activity of most non-proofreading DNA polymerases. Now, this insert also contains the A overhang. If you do the PCR using a proofreading polymerase, then you must perform adenine tailing on your PCR product. This is achieved using DATPs and a special clenau that lacks the exoactivity. This tailing will add an adenine to the 3' end of the PCR product. This A overhang insert is now ready with its 5' hydroxyls to attack the phosphate on the vector. The A will base pair with T and the nucleophilic substitution will join the vector and insert. Once again, this is a circular final vector that is partially ligated, and the remaining nicks can be fixed once this vector is inside the bacteria. In this vector, there is no self-ligation issue, because vector closing depends on the sticky base pairing. T cannot base pair with T. Let's take a small detour. You can do this with restriction enzymes as well. Essentially, you need a T overhang vector and an A overhang insert. The T and A will base pair, and ligases can be used to ligate the insert and the vector. This is called TA cloning because of the pairing between T and A bases. This is a non-TOPO version of TA cloning. Hopefully you see why this is called TA cloning. The big issue here is that the product is symmetric in all TA cloning methods, meaning that you can flip the insert 180 degrees and there's a chance that you insert this reverse orientation. So, similar to blunt TOPO cloning, you have to screen for your desired vector in TOPO TA cloning as well. The last strategy is the TOPO directional strategy. Simply, this implies that the insert can only be cloned in one specific orientation. Like other TOPO vectors, it is also linear, but instead it is an expression vector. Typically, you would use this for cDNA or gene cloning. Note that I mentioned how this vector uses both sticky and blunt sites. One of the vector's ends is a blunt end. This other end has a sticky overhang GTGG. The insert in this cloning is typically blunt. You can get it from type 2S digests, but typically you don't do that. Almost 100% of the time you have a cDNA amplified from PCR that needs to be cloned. And PCR amplicons from proofreading polymerases are blunt. Now, let's say you want to insert this DNA in the X to Y orientation, meaning that X corresponds to the sticky side, the Y corresponds to the blunt side of the vector. So when you do a PCR, you must add CACC as the 5' overhang to the primer on the X side of the insert. Here is the final corrected form of the insert. Note that this GTGG in vector is in the same direction as the GTGG on the insert. So when you mix this vector and insert together, the Y side of the insert can attack the blunt Y side of the vector. But the X side of the insert cannot attack the X side of the vector because there's a sticky overhang. So somehow the hydroxyl needs to be brought together, closer to the topo in this situation. How is this achieved? Well, CACC is complementary to GTGG. This GTGG from the vector can strand invade into the blunt X side of the insert and try to base pair. As a result of this strand invasion, the GTGG of the insert is kicked out. This brings the C and T close to each other. This allows the hydroxyl from the C on the insert to attack the phosphate on the T from the vector, and the two DNA ends are ligated. The other side is just blunt, so ligation on the Y side is not a problem. Now we have a partially ligated circular vector which also has a small flap on it. But again, this is not a problem. The bacteria has flap endonucleases that can cleave the flap and seal the nicks. You may have seen this type of flap cut and nick sealing in Okazaki fragment maturation in bacterial DNA replication. So this way you can clone the insert in a specific direction. I hope it is obvious that only one side of the insert should contain GTGG. Also, it should be obvious that there is no self-ligation issue in this vector. One last point about what makes this directional vector an expression vector. The vector contains some sort of promoter like sp6, T7, or maybe the pBAD from the arabinose operon. This follows the ribosome binding site which lives next to ATG, our first codon. In frame with the ATG codon is the CCC from the cloning site. The next codon is then TTC and ACC. This means that the GTGG overhang of your PCR primer is already part of a codon in frame with the vector. And this means that the first base of the insert, the N, is the start of a new codon. And this continues until the last nucleotide, which ends in a full codon. After the N, you start with the AAG from the vector, which is a codon, and this continues till a stop codon. In between, you may have some purification flag tags or something else. Depending on the promoter in the front, you may have some specific terminators or poly-A signals at the end of this expression cassette. So this way you can use the cloned insert, which is a template for making RNA or even proteins from those RNA. If you found this video useful, I have many more videos on cloning and genetic engineering in the playlist linked in description.